about biotechnology, vaccine, drug development and so on. This year I have chosen a topic, synthetic biology. Some of you might have heard. It's a technical subject actually. I'm not very comfortable with the subject myself because, as Dr. Amrao said, I didn't like engineering, I went into chemistry. Now I realize I should have known little more engineering. Because synthetic biology is actually application of engineering principles to biology. This is the definition of synthetic biology. Application of engineering principles to biology. And you will see, and it's a fairly recent subject. It's not even 10-15 years old actually. Although the background we know, several molecular biology giants have contributed. But it's exploding. The subject is exploding, and I find many of our students are not even aware of this subject. I will mention it, what is happening internationally and where we are in this subject and what is the potential of synthetic biology. And that is my, the biology part I am very comfortable, the engineering part I am not that comfortable, but I thought I will give you the flavor of this subject for those who are not familiar. Some of you might have heard about this field as well. Before that, let me give you, synthetic biology today has evolved deeply with microorganisms. Although it is moving into mammalian cells, higher organisms, cells of higher organisms, but till recently it has developed greatly with microorganisms. And what is the utility of microorganisms? I think this is where the biology part becomes very important. These are the model systems to understand life processes. At one stage, we all used to believe what is true of E. coli is true of elephant. E. coli is a bacteria, as we all know. But many of the processes in bacteria do happen in higher organisms, but the complexity is much greater in higher organisms. But even today, bacteria is served as model systems to understand life processes in the human being. We will more concentrate on application. And bacteria are used for many production which have application in health and disease, food and agriculture, environment and industry. Some of you may not know, microbes are the most abundant living systems in the globe actually. There are 10 to the power of 30 organisms in the ocean and the soil, and equal number in the ocean. That is the number of organisms that are present, but we know of only less than 1% of these organisms. That is because we only know organisms when we are able to culture them. You have to grow them in some media and then say this is E. coli, this is Streptococcus faecalis, this is pneumonia, all that we will be able to say. But the soil and oceans have organisms which we have not cultivated. We don't know how to cultivate them. But how do you know they are existing? By DNA analysis. Taking a sample of soil, take a sample of water, or sea water, and analyzing the DNA, and today technology is available to identify various combinations of DNA that is present. On that basis, you can say so many organisms exist. The subject is known as metagenomics. This subject is known as metagenomics, identifying organisms based on DNA sequence. Organisms which you cannot see, organisms you cannot cultivate. Till in such time, you have got a condition in which you are able to grow these organisms. But this is the number that is available in the globe actually. What also you need to realize, if you have a bacteria and the bacterium grows, and theoretically you assume there is no limitation of medium. You provide all the nutrition which the bacterium needs. And E. coli, for example, has a half-life, doubling time of 30 minutes, let us say. It is calculated to produce a population with the mass of Earth in 4 days. If you allow bacteria to multiply uninterruptedly, theoretically providing, medium will deplete, but you assume you are able to provide all the media. In 4 days, it will produce size of mass of Earth. That is why I am saying all this, that shows the advantage of these organisms to be exploited for commercial purposes. That is what we are coming to. Few examples in health and disease: probiotics, antibiotics, anti-tumor agents, immunosuppressants, they all come from microorganisms. In food and agriculture, food processing, bioorganism, biopesticides, bioremediation in environment, bioenergy is the big subject, biocatalysis, bioconversion. 500 commercial products are made using microbial enzymes. So the potential for application is huge. A few examples, let me give you: industrial enzymes, organic acids, fine chemicals, antibiotics, microbial insecticides, anti-parasite agents, vitamins, amino acids. This is just a tip of the iceberg. But so many products come from microorganisms which probably we are not aware of. Microbial technology has evolved. Classically, people know you culture an organism, you can isolate some products, and I have given you some examples. But the science has developed enormously because with DNA technology today, recombinant DNA technology, fermentation process, high throughput screening, strain improvement, bioreactor design, downstream processing, cell fusion, metabolic engineering. Some of these terms may not be very familiar to some of you, but basically these are technologies by which you can grow organisms in a reactor, bioreactor. Just like chemical reactor, you can also have bioreactor. Grow organisms in 10,000 liters and produce whatever you realize. And all the technologies that are needed for this, you have them. Now comes the subject of metabolic engineering. What is metabolic engineering? Now, organisms produce an amino acid. Certain amino acids in nature you can isolate. I have given you examples. But you want to change. You want to produce something which the organism is not producing. You want to force the organism to make what you want. That is known as metabolic engineering. The example I give you is this. Many of you would know about this plant which makes indigo. Indigo is a dye, blue dye, as you know. There is a lot of, Gandhiji has written a chapter in this book on my experiments to truth. He has written on indigo, how the indigo farmers went through a lot of problems with the British and so on and so forth. Now, depending on the plant to make indigo, can you make a microorganism produce indigo? That is an example of metabolic engineering, and you have to play around. That is why I said it's a technical subject. I hope there are boys and girls who have studied a little bit of molecular biology and biochemistry here. Tryptophan is an amino acid. Tryptophan can become indole. Now, indole can become indole dihydrodiol, but indole will form from by E. coli, this organism. But this step, this organism cannot do. So you take this gene from another organism and put it here. E. coli cannot make this product. But you take this enzyme from Pseudomonas and put it here. Now you have come to indole dihydrodiol. Now what you do? You want dihydrodiol, spontaneously will give you indoxil. Indoxil on air oxidation can give indigo. But what happens is, indoxil can also, there is a side reaction, it will form isotin and isotic acid, which will undergo air oxidation and give interleukin, which is burgundy. You don't want burgundy, you want indigo. Therefore, what you do is to degrade this isotic acid with an enzyme, acetyl hydrolase, and this is from another organism. You don't have the remember the pathway and so on. What is being done here? E. coli can produce indigo. If you engineer this organism in such a way, the pathway is altered, you get a gene from some other organism and put it and block the side reaction and you get what the product you want. This is known as metabolic engineering. Now you can realize what metabolic engineering can do. You want a unsaturated fatty acid, in a coconut has got the saturated fatty acid. Let's say you want to make unsaturated fatty acid. You can put a gene from some other organism and make it. Whether actually it is, feasibility will come to it later. Theoretically in the laboratory it is possible. In fact, you can produce anything you want like that, using metabolic engineering. Now, metabolic engineering, the next revolution has become synthetic biology. I have given you all the background to give into the subject of synthetic biology is even more complicated than metabolic engineering. Why it is even more complicated? I must also acknowledge there are huge number of papers. These are the most recent ones from Nature Reviews in Microbiology, Nature Special. There are huge number of articles. I've been interested and I've read some of them. We want to get into at some point of time in research. I don't know how much time I have. I'm 76 only, so I'll try. See, the foundation for all this is where I said it's a technical subject. It's based on molecular biology, development of molecular biology. So you have to have a background in molecular biology to get into this area of synthetic biology. That is one part. The other part you have to understand are the engineering principles. I will come to the engineering principles in a short while. So, the founding of this synthetic biology was almost near the turn of the millennium. That means around 2000 years. 2000, this is where the whole subject got a fill. And a group of engineers, physicists, computer scientists, along with molecular biologists, said we will join together and see what we can do. This is how the whole thing started. Now, genomics revolution, rise of systems biology. I will not get this other subject, systems biology. I hope somebody else will talk about it. The synthetic biology has undergone dramatic growth in the last decade or so. The aim of the synthetic biology is to predictably bioengineer organisms that perform beneficial functions, from producing antibiotics to purifying contaminated water. I have a slide which says sky is the limit. You can do anything you want. If you are smart enough, you can achieve. That is where, using just microorganisms, it is possible. Although this has now started being applied to higher organisms, mammalian cells and so on. But the idea was not to make application. The idea is to understand, we still do not understand how our cells behave. Despite all the discoveries, Nobel laureates, Nobel prizes, we don't. So we have this phenomenon, what we try to do is reverse engineering. So we try to do certain things outside, assuming this is probably the way our cell is behaving. Then do a lot of research and find out cell is not that simple. It is even more complex than what you do. So to understand reverse engineering was done to understand cell behavior. But in the process, applications are tumbling out. I have to get a little more technical. Here, this is a very fundamental thing in molecular biology, lack of hormones. Let me say, for those who are not in life sciences, it is simply a switch mechanism all cells have. A switch followed by a set of genes. The genes will produce enzymes or proteins. If the switch is on, all these enzymes will be made. If the switch is off, none of the enzymes are made. This is known as operon. This is a simple example of the lack of. This is a classical Jacob-Monod model for which they got Nobel Prize. So this is the regulator which produces a repressor which will go and operator. If the operator is off, all these genes genes are not produced. The operator is on, all these genes are produced. Now, how this on-off mechanism takes place? On-off mechanism takes place because it produces the inhibitor. We call it the repressor protein. The repressor will go and sit here and turn off the switch. And then these are not produced. But you add sugar like lactose, it will reverse the effect of the inhibitor, and therefore this will not be blocked, and now all these genes will be made. The switch is on. That is, this is where the repressor cannot be made, or it is annihilated because you have added sugar lactose, and therefore the switch is now in an on position, and all these genes are made. If you have a tag here, what we call is a fluorescent protein, now the cells will fluoresce. If the cells fluoresce, you know the switch is on. If the cells do not fluoresce, you know the switch is off. The on-off mechanism, that is trying to get it from the engineering principle. They call it as gates, 0, 1 in computer language you all know, low voltage, high voltage, 0 volt, 5 volt. Whether it is, or they call it a true or false, then there the entire circuitry behaves in electronics actually. But it's not just like operon. In the last 3 decades, central dogma, DNA gives RNA, RNA gives protein. Then you can clone any gene. You can put a mammalian gene in a bacteria. You can take a plant gene and put a bacteria gene can go into the human being. You can play with these genes and then they can express in different environments. I have given lectures on this earlier. You can make human insulin in the yeast. Bacteria can make human insulin, and then vice versa. So all these things are possible. Then comes PCR amplification. All the time now getting DNA, getting the gene used to be a huge problem. Now this reaction came, now you can make the DNA in a test tube. In a 1 hour time you can make a large quantity of DNA in a test tube. Polymerase chain reaction. I will not get into the mechanism, or another person got a Nobel Prize. Then it went to what you call the evolution of omics sciences. This also I have said before, the most important omics, we call it as genomics, proteomics, metabolomics, but somebody said the most important omics is economics. But in spite of that, what is the difference? In my career, we used to study 10 genes, 15 genes, 10 proteins. It's a big achievement. Now nobody will look at. We want to look at all the genes in the cell. 25,000 genes are there, I can look. I can put all the 25,000 genes in 2 by 2 centimeter chip. This is what the field has come to. If you are talking about 100,000 proteins, I can analyze all the 100,000 proteins. These are called the omic sciences. You are not looking at a compartment. You are not looking at 1, 2, 3. You are looking at the entire gamut. In diabetes, if you say, how many genes are different between a normal cell and a diabetic cell? I will say 300 genes are different. This is because of microarray. Because I can make a microarray from a diabetic cell, I can make a microarray from a normal cell, and I can find out which are the RNAs which are expressed in the normal cell, which are the RNAs which are expressed in a diabetic cell. And I can come to the conclusion that 300 genes are involved in diabetes. So this is how the field has moved. That is called the omic sciences. Now it has gone all the way. People are now trying to synthesize a cell. A living cell can you synthesize? Don't ask scientists are trying to play God. This is there, the field is moving. I'm not going to talk about those. I'm only talking about this. What is this thing which few years ago, 2010, Craig Winter, he's a crazy guy actually. But phenomenal, all these guys are crazy, but they are phenomenally intelligent. He said, I'm going to synthesize a living cell. Of course, it's a they didn't really synthesize the living cell, they are far, far away from that. But what he did? He did what he took the small organism called the microplasma mycoids. Because today DNA synthesis, DNA sequencing has become child's play. I should also tell you, there is a human genome project. People wanted to sequence the entire human genome. $3 billion and 15 years was allocated for this. So many groups all over the world, groups were formed globally to understand the human genome sequence, which consists of so many 26 chromosomes. But it was done much ahead of time actually. And the cost of DNA sequencing is coming down and down and down. But even the microorganism, to do the DNA genome sequence of a microorganism used to take, say, 1 year, 1.5 years. Today, one microorganism can be sequenced in 1 week. DNA of a microorganism can be sequenced in 1 week. This is how the field is moving so fast. So what he did was, knowing the sequence of this mycoplasma mycoids, he did isolate it from this organism. Knowing the sequence, he synthesized it in the test tube. Therefore, it is totally synthetic DNA. It is not, it is man-made DNA. Of course, knowledge has come from the organism. He took this mycoplasma mycoids, DNA, and put it in mycoplasma capricolum. Now, he destroyed the DNA of the mycoplasma capricolum, but put this DNA into this organism. And then he showed this organism can replicate. And what replicated was this DNA and not this DNA. The claim is it's a totally synthetic DNA. The totally synthetic DNA has been taken from one organism and put it into another organism. The DNA of the second organism has been inactivated, so it cannot replicate. And what is replicating is the synthetic DNA. So everybody said, oh, the cell has been synthesized, synthetically made and all that. You still realize he used the receiver, the receiver is the second organism. That is still very, very important, and we cannot still make those kind of things. And he went all the way to show it is really synthetic DNA. And he synthesized this DNA, which is what we call 1.08 megabase. That means 1.08 in 10 to the power of 6 base pairs it has got. DNA has got 4 bases, A, T, G, C. The A, T, G, C is present in various combinations, million times in this organism. It is the smallest genome containing living organism. Therefore, that is why he has chosen this particular organism. And he synthesized it, I will not get it again. First he took 10 base pairs, 100 of them. Then he took 100 base pairs, 11 of them. And then he put them all together. Synthetically everything was done. And this is all, it describes how the whole thing was done. And this is really interesting. To make sure people should not say, this is not true. Your DNA, what you put, did not replicate. So he added watermarks, as he called it. He introduced a poem in the sequence. To live, to earn, to fall, to triumph, to recreate life out of life. He has quoted it in the DNA. What I cannot build, I cannot understand. Richard Feynman. You put that sentence also in the DNA. You may be wondering, how do you put those sentences in a DNA? DNA has got only 4 bases. So, amino acids have, English has 26 alphabets as we know. And there are 20 amino acids. Now each amino acid we have a code. When you say lysine, you will say it is K. When you say tryptophan, it is W. Therefore what he has done, for each one of these, for example, when you say W, he will put the tryptophan codon there. H is histidine. Histidine has got a codon, three bases. He has put there. So 6 of them you still have to, then they can put it if it is a non-coding sequence, it can. this is, just by design, to make sure people should not claim that it is not a synthetic DNA that is replicated. So he actually showed the replicated DNA contains all these watermarks. And he has put the addresses of all of his students in the DNA. All these things he has done. Very interesting. So, having gone, so what we have started? We have started with a switch mechanism of operon. There you can turn on, turn off genes, to omic sciences, to capacity to synthesize DNA, which means you can synthesize huge amount, big DNAs you can synthesize. You can make them replicate in the test tube. Put them in the organisms and make the organisms replicate. These are the capabilities of molecular biologists today. Now comes the synthetic biology part. Now how do you make use of this knowledge? They want to create circuits now, biological circuits like electronic circuits. This is where the synthetic biology challenge is today. Now, this is where I found I should have studied a little more engineering. Now, digital circuit, elementary, a logic gate. The logic gate is what they call the 0-1, basically. On, off, true, false. And I understand there are several logic gates, A and D logic gate, OR, WR. That means you may have two inputs, input A, input, two inputs will give you one output. Sometimes only one output can give you this, the input can give you the other output, the other one can. And sometimes this either or. All these logic gates are available today. This is how the principle I understand is used in making electronic circuits. And many such logic gates constitute a microprocessor device. Now can we do it in biology? This is the challenge of synthetic biology. Using combination of logic gates in a microprocessor assembly, you see they can be arrayed together, integrated circuits are made. How many circuits, how many you can accommodate depends on space, constraints and so on. Now, here our workhorse is E. coli, bacteria. Remember that, So what are the parts of engineering to make a machine? Electronic, wire, transistors, valves, diodes, relays, electronic switches, etc., etc. Now, what are the parts of the synthetic biology? Genes, RNA, proteins, manipulated through transcription, translation, post-translation modification, and so on. These are the parts of. Okay, so equivalence. This is how one wants to. Now, what is an example of a simple design? I don't have time to explain each one of them in great detail, but I don't think you will be able to see what it is conveying to you. A switch will be turned on or off. I gave you the example. That is linked to a fluorescent protein. Whether you can have one switch, you can have two switches. How much behavior? You can have a behavior of fluorescent going up, going down. You can have a bimodal function. You can have one forward switch, one reverse switch. You can have three such switches to create very many different patterns. I think if there are students of molecular biology should take each one example and analyze how do you get the final output. It is basically different switches which can be turned on or on, connected in a circuit. And see what would be the ultimate behavior of this cell. This is what they have done so far. I hope you are able to see this IND gate, for example. Input A, input B, then the single output. And these are examples, very many examples where one switch, two switch, three switch, four switch, and you can get patterns like this. Depending on the reporter, if the green fluorescence or the red fluorescence, you can have beautiful patterns like this. Again, let me repeat. The whole concept of digital electronics, using valves, diodes, etc., is being replicated in biology using genes, proteins, RNA, and so on. Turning on or off a switch means here the compound, a molecule is produced or not produced. And you are not doing one molecule. This molecule will act on the second switch. That molecule will act on the third switch. You can turn off or turn on these switches using all play around with molecular biology tools which I have told. Those tools are what I have described earlier. You can synthesize, if you make a circuit which may be 100 kilobase of the size, if you make a circuit with 5 switches, 6 switches. Today, the entire 100 kV can be synthesized in the test tube, which is not possible dedicated. Using that now, what they have done, they have put all these and then made an antibiotic called virus. This is an antibiotic which they are able to make. You can see one acting on the other. These are the various steps in making it. A single step they are making this compound. Too much light, but this is the one that has really been successfully made today called artemisinin. Artemisinin is an anti-malaria drug. Probably the only drug that works today globally. And it's the most important drug. Many of the other molecular etc. have become parasites have become resistant. So artemisinin is made by a plant called Artemisia annua. Now, I told you, they gave the example of E. coli trying to make indigo. Now, can we make artemisinin by E. coli? Can yeast make artemisinin instead of going to the plant? It's a very complicated molecule. Artemisinin to make, you need so many steps. And all these are not present in the yeast or the E. coli. So they take the genes from the plant, genes from this organism, genes from this organism, put them in a circuit. That is where the synthetic biology is working. They have put all these and created this circuit and made artemisinic acid and converted artemisinic acid to artemisinin. And it is not a laboratory marvel. Now it is commercially viable. They are now making 25 grams per liter. They have scaled up the whole process. Now they say we will now make a artemisinin in a fermenter. Of course, now there are sociological questions. Internationally there are several farmers who are growing this plant to make artemisinin and they are selling. They are asking what will happen to these farmers. So everything has a side reaction. We have to worry about. Scientists do not realize. We think we have achieved a great deal. 20 grams. Now if it is started, Roche or big company will take that thing, and all these farmers will be in the end. They have to do some other profession. In China, Korea, even in India, there are people growing this Artemisia annua plant to make artemisinin. But for synthetic biology, it is a great success that you have genes from different organisms and put in one particular organism and then make them to work to make the molecule. So this is a circuit which is very similar to what can happen in digital electronics. There the output is different. The simple thing which our even not so well educated people do, when the current passes, you get green light, blue light, after a few seconds it will turn off again, it will start going. These are all on-off mechanisms. Same on-off mechanisms are being applied here. This is what they have done. They have got something like 25 grams per liter of artemisinic acid. Sorry. Using this technology, they are now wanting to make so many other molecules. Vincristine, morphine, prostatin, bleomycin, paclitaxel, citric acid, anti-cancer drugs. Many of them are anti-cancer drugs. Can we make anti-cancer drugs using microorganisms by synthetic biology? So, recent success, it has even further gone further. They want to now make isobutanol, fatty acid-based biodiesel, gasoline and bioplastic, 1,4-butadiene. So you take amino acid, you take sugars, can you convert them into gasoline? Can you convert them into butadiene? All that you need is two reactions, you have to chart out the reactions. What are all the reactions? The organism will not be able to do this. It may have only one gene for that. But all the others you have to source from this organism, that organism, you have to put those in the system in here and create a circuit. And then see whether you succeed. I am giving you the companies, number of biological systems, designers, manufacturers. who are doing research company level, home and personal care products, flavors and fragrances, paints and coatings, cosmetics, adhesives, lubricants, agrochemicals, pharmaceutical intermediates, animal feed, belong, biocides, urban pesticides, antifungals, and just reading them. These are the kind of molecules which they want to make, companies want to make using synthetic biology. Which means anything you think of, any molecule you think of, you can make, theoretically. More applications. Can you take a virus which causes a disease? Now you can modify the virus in such a way that it cannot produce the disease, but it can act as a vaccine. Sky is the limit. They want to recreate extinct organisms, the Spanish flu virus. I'll come to the ethical part in the last slide. Why do you want to revive? Somebody wants to revive the smallpox organism. The reason being, if you revive them, you can now find out a drug. Find out a drug for an organism, the Spanish flu, which had disappeared, which caused havoc in 1918 or something like that. But you revive them and so that you can do research and find a drug and keep it ready when such a thing eventually happens in the future. Synthetic photosynthesis. We know plants can do photosynthesis. Now can you make photosynthesis happen in a test tube? Can you make biofuels from sunlight? Basically reinvent nature and ourselves. It has become, the science is exploding. Lot of hype, I can tell you. We are not, other than artemisinin, they have not produced anything in a commercial level. Everything is in a test tube, everything in publication and so on. It will take time. But you see, thinking process is very, very important. How things are moving so. Now this is the synthetic biology in mammalians. All these I talked only in bacteria, microorganisms. Now they are moved into higher organisms, cells, human cells. Can you do synthetically modify human cells? Then, they have shown up several applications. Synthetic biology leading into therapeutic interventions are possible. Cell-based therapies are possible. Protein drugs are possible. Vaccines are possible. Gene therapy is possible. What are the obstacles? It's easy to say you put all these together. This is where what realizes engineering works in isolation. You can put all these valves, transistors, and you can predict this is how it will go in a cycle. It will work. There is no external influence to affect this. But in biology, what happens? You put this circuit and put it in an organism, it will not work. Then you start asking, why it didn't work? Individually, each part worked. When I put them together in biology, it did work in this organism because there are millions of other compounds within the cell. And what is the influence of those compounds in this reaction, in this environment, you don't know. See, our cell, living cell, contains millions of compounds. Body makes. You may say my lactose should repress, arabinose should induce. E. coli will do. When you put the reaction in a mammalian cell, it will not work. Then you will find some other molecule is interfering with this. This is the problem which the synthetic biologists are facing. Contextual. What individual parts are working in the test tube so nicely, when I put them together in a different environment, it's not working or not working to the efficiency. Therefore, they have to redo everything. This is where the biggest problem they are finding. How to make it functional and how nature is an optimized process. And the other part is part standardization. You have C certification in industry. You have ISO certification. How do you certify a gene? How do you certify an RNA, a DNA? How do you do that? Because these are all the parts. Can you do that? So people have started this already, part standardization. Now there is an organization that is available. I want this gene, this enzyme, this thing, they will supply. This is happening internationally. abstraction hierarchy. Huh? This means I didn't know what it means. And then I found out. It is basically modular. There are several modules which have to be assembled, to make a product. And these modules are now available today for those who want to test. I want to make some compound of my own. I want to use this organism. What is all that is needed? What are all the parts that can go into that? Similarly, there is a registry of standard biological parts. Like every part, if you buy a relay, these are the dimensions, these are the parameters for this. Can you give such things to biological components? Because our components are genes and proteins and RNA and so on. This is happening. And each one is being looked at. BioBrick we call it. And the biggest thing that I think the students should notice, this is the one. They have started an international genetically engineered machine, which is an undergraduate synthetic biology competition. This has been started in 2004. No? Let me read this again. International genetically engineered machine. Any student can take any organism and say, I want to put all these genes together and I want to make this product. I want to make a new organism. And this has become a competition. And all these ideas are being thrown at young students, who will develop all this. MIT started, these are the guys, Jim Collins and all this from MIT who started this. Bring researchers from biology, chemistry, physics, engineering and computer science. This is what is happening in this field. And identify a community and galvanize efforts towards the design, construction, characterization of biological systems with long-term goal of whole genome engineering. So the power of youth is very important here. This is what they are doing. IGEM, International Genetically Engineered Machine. This is an undergraduate synthetic biology competition. It has been taken very seriously all over the world today. You will just see what is happening in India. Anyway, this is the history which I talked all along. Just you think it is all laboratory marvels. Look at the companies that are involved in this field. Cargill, DuPont, Pfizer, Virgin, Amaris, all these companies are involved, basically cutting, testing the DNA, getting the gene, creating biological substitutes, and each one is interested in their own field. So this field is impersonal kind of thing. You can use it for anything. You can observe light and want to use the light. That's why they are talking about synthetic photosynthesis. That means you don't have to depend on plants to do photosynthesis. You can do photosynthesis in a test tube. Where is India? This is what I want to end my talk with. US has been a leader, as usual. Other countries, including super economy of China and some with collective entity of European Union, are investing heavily in this field. And this was the global synthetic biology competition undergraduate students in, I think, 2011. 2014, this undergraduate thing is going to be held in Israel, I think. Some 246 teams are participating. Undergraduate students trying to do synthetic biology. 82 teams are from Asia. Of which, of the 250 teams, 82 teams are from India, are from Asia. And I was looking for India here. I found actually only one. Rest 80 probably from China. Or at least 60 from China. You can see, China, Taipei, China, everywhere it is China. We have one team out of the 252 teams in the world from IIT Delhi trying undergraduate competition in synthetic biology. This is where, are we missing the boat? Not that, tomorrow it is going to solve all our problems. It will not. It's an exciting field. It's a field where physicists, engineers, computer scientists, biologists coming together, trying to create circuits to make a product, and it's not going to be easy. I told you, biology is non-linear. It's not like other subjects. You may show all these individual reactions working beautifully. Put them together in a new environment, it will not work. then you have to find out why it is not so. At least parts are getting standardized. Now people say, you want this gene, okay, write to us, we will give you some databases are available today. There are people, not all genes, but if you are lucky, you will get it. This is my last slide. Now this excitement, as scientists, we tend to forget the regulatory issues here. As usual, people are concerned. Insufficient basic knowledge about potential risks posed by design and synthetic organisms. What kind of harm it can create to the environment? People are telling, I will revive the smallpox flu. People are telling, I will revive the Spanish flu, which caused the flu in 1918. What is the consequence of that? Suppose it escapes into the environment. What will happen? Uncontrolled release, accidental or deliberate, of novel genetically modified organisms with environmental or human health implications. Bioterrorism, biological warfare, construction of novel hostile organisms it's also possible, theoretically. I can create an organism which can produce poisons and release it into the environment. No, this hacker culture in computer science is so there can be biohackers. While you are doing, you think somebody may hack. Hackers will come and take up unregulated individuals developing dangerous organisms. Patenting of genetic material, and another one is the multinationals will be leading and then they will be patenting all these organisms. What the hell does it lead to? Trade and global justice destruction, creation of artificial life and related philosophical religious concerns. Now when you say at the moment I told you, our scientists thinking they are playing God or something, they are wanting to create this, who are these people? Like this, just those are issues. So, my worry in our country is they are very good at the armchair ethics. Using them to not to do anything. This will happen, that will happen, so don't do anything and suffer. Millions will die of tuberculosis, but millions will die of starvation. You bring a technology which will do, no, this technology is dangerous, don't touch it. But millions can die in the meanwhile. This is a theoretical assumption, it will be harming, and don't let it go. This is our armchair, ethical, we are all highly ethical people, to let people die so that nothing happens to our value systems. So, but keep this ethics at the back of your mind. Those who get into the field will say the NGOs will come, they will be just waiting for this opportunity and then say, oh, this is very dangerous, you are creating an organism. But one biggest advantage why I spend time on microorganisms is you can grow them in a contained environment. You can grow in a factory, a bioreactor. There are many, already we grow many organisms, we produce amino acids, vitamins, all these organisms are being produced. So we can create harmless systems and very, very many useful products can be made. It's only ingenuity in your field. People should think, what is the molecule which I have to make? Why should I depend on the plant? Why should I depend on the animal? Can I make the organism to make microorganisms which I can grow in a culture, which I can grow in a bioreactor and isolate these molecules in large quantity? That is where the excitement is, I think. Thank you so much.